The cell cycle. The cell cycle is an ordered set of events resulting in cell growth and division into two daughter cells. During each cycle, cells go through a series of 4 phases: G1, S, G2, and M. G1, S, and G2 occur during interphase, the period of DNA replication, which is followed by the M phase, also known as mitosis or cell division. Some cells in G1 are not ready to progress in the cell cycle and enter the 5th phase, G0, a subphase of G1. The cells in G0 remain indefinitely until they're ready to proceed. Some cells may never leave the G0 phase. Others, such as bone marrow cells, may skip the G0 phase entirely. Cells begin the cell cycle by entering interphase. In interphase, the cell prepares for division by duplicating DNA and making necessary proteins. In the G1 phase, the cell prepares itself to duplicate its DNA. The synthesis of RNA and protein also take place. The initiation of replication of DNA marks the transition from G1 phase to S phase or synthesis phase. The total content of DNA is duplicated into 2 exact copies. In G2, RNA and proteins that are necessary for cell division are synthesized. Proofreading also occurs. The DNA replicated in the S phase is checked for mistakes. If mistakes are found, the cell repairs them or receives a signal to self-destruct called apoptosis. In the M phase, the cell undergoes the process of mitosis and divides into 2 identical daughter cells. Mitosis is separated into prophase, metaphase, anaphase, and telophase. In prophase, replicated chromosomes condense and then split into identical halves called chromatids, which are held by the centromere. In the cytoplasm, the centrioles replicate and move to the opposite poles of the cell, and spindle fibers form. As nuclear membrane and nucleolus disintegrate, the spindle fibers move to the area where the nucleus was previously located. In metaphase, each chromosome, consisting of 2 chromatids, attaches to spindle fibers via the centromere and move to the midpoint of the spindle, the equatorial plate of the cell. In anaphase, the centromeres split and chromatids are pulled to opposite sides of the cell. When chromatids reach the poles, anaphase ends. Each sister chromatid is considered a new chromosome. In telophase, the chromatids uncoil and return to the interphase form of 46 chromosomes. The nuclear envelope forms and nucleoli reappear while spindle fibers disappear. Cytokinesis, the cleavage of cytoplasm, occurs, and the cytoplasm divides into equal parts. 2 identical daughter cells have been formed. The newly formed cells are ready for another round of cell cycle.